Great. Hi, Perry. Welcome.
Good morning. Hello,
Great.
everybody. Hi,
Great.
Angela. Hi, Sai.
So good morning, Phil. Good afternoon, Phil. It's two.
Yes,
Hi,
lovely to
Barry.
see you, Stu. Afternoon indeed.
Fantastic. Okay. Well, I'll go ahead and give my introduction, guys. Welcome. This is Agential Topics. I'm Charlie Munford. Agential Topics is sponsored in part by Talking Octopus, a Vermont nonprofit. And on the blackboard of Richard Feynman, on the day he died, was written the words, "What I cannot create, I do not understand." And so, Talking Octopus is organization dedicated to building a physical model of biological agency. And the Agential Topics is the study group where we get together and try to figure out what biological agency is. So, today, our guest is Philip Ball, science writer and one of the foremost explainers of biology. And with very little further ado, I'll just let Philip begin his presentation for us today. Thank you.
Thanks so much, Charlie. Right, let's see if I can get the screen share up and running. And then I guess
Perfect.
make that full screen. Okay, that hopefully that looks good. Okay. Um,
Perfect.
I can skip past these introductory aspects pretty quickly, I think, with this group. What I wanted to point out in just introducing the idea of agency is really that these very mainstream guys, Ernst Mayr, evolutionary biologist, Jacques Monod, a molecular biologist, really, have both embraced the idea that and made it very clear that purposive behavior and goals are a fundamental aspect of biology. And, Mono goes as far as to say that one of the fundamental characteristics common to all living beings, without exception, is that they're endowed with a purpose or a project. And that, to me, is the defining feature of agency, which I would suggest thinking of it as this way. It's the ability to manipulate oneself, one's environment, in order to attain some self-determined goals. And this capacity means that organisms aren't automata that just respond to the environment, maybe through some kind of internal lookup table, some pre-programmed set of actions triggered by particular stimuli. Instead, they are improvising. They're using heuristic rules of thumb. They're revising decisions as circumstances change. So agency has this fluid, contingent, approximate, not rigidly rule-bound character to it. And again, it probably goes without saying to this audience, but nonetheless, I think it's worth stressing that invoking agential behavior, in my view, in no way conflicts with a reductive approach to biology. We can consider an agent to be acting on its own behalf, in some sense, while recognizing that its behavior can be broken down and analyzed that way, can be looked at reductively. There are modules that would give rise to it, and the question then is, what exactly are those reductive roots? What are the molecular principles underpinning agency? How can living systems achieve responsiveness to their environment that's adaptive in the behavioral sense, that's contingent, that's even innovative? So that's really what I want to explore. And this is really an exploration. I'm thinking about these ideas, but I don't have answers. I just have a few things to suggest, to throw out to you for discussion. So in the paradigm that Crick's central dogma implied, information flow in biology is regarded as a bottom-up process that begins in the genome. And the invitation was to suppose that the specification of the organism and its behaviors was somehow inherent in this specification of a set of proteins, which somehow conspire to produce the phenotype. So in that view, not just sequence information, which of course is all Crick was talking about, but all causal information in biology flows up from the genome, and more specifically from that part of the genome in this picture that is concerned with encoding protein structures. So that picture, I think, is obviously wrong. Biological information flow generally does not have that bottom-up unidirectional exclusivity. The morphology is not somehow encoded in the genome. The informational systems, the informational flow in living organisms. So it isn't just this step going to the phenotype. The informational flow is omnidirectional and open. Although the central dogma obviously summarizes a significant part of that network of information flow. So this kind of openness is indispensable to agency. What's more, it's worth remembering that even within the central dogma itself, in the strict definition, molecular function itself is not, in general, fully specified by structure. It's created too by context. One example of that might be in the ability of some proteins to moonlight, as it's called, to perform very different roles in different circumstances. This might be regarded as just the sort of opportunism of an improvisational evolutionary process, but I think it surely reflects too the way in which nature seems to produce molecules with potential behaviors rather than machine-like parts with highly specific and constrained roles. So some proteins have functions that are left very much open to influence, thanks to their ability, for example, to bind to many different ligands. The transcription factor P53 is a great example of that. It binds to many different DNA motifs, it binds to many different proteins, and that gives it roles in a large number of cell processes, from metabolism to the cell cycle to DNA repair. It's often spoken of as a tumor suppressor, but I think in truth it makes little sense to speak of a molecule like P53 in terms of a function at all. One might better think about many biomolecules like this in terms of affordances, of the possibilities for action that that molecule seems to possess. And this view undermines the idea that the genome is a blueprint for the organism. I like the way that Evelyn Fox Keller spoke about the genome. She said today's genome looks very different from the one with which the science of genetics began. Rather than a set of genes initiating causal chains leading to the formation of traits, it looks far more like an exquisitely sensitive reactive system, a device for regulating the production of specific proteins and other molecules in response to constantly changing signals it receives from its environment. So I'd suggest that we might then think about the genome simply as this, as a resource, as a set of resources that cells can use, and in particular, as a source of functional molecules, including molecules that have just the potential for functionality that may not be tightly specified in advance. And a view of the genome like this makes no assumptions about biological causation, and I think it can restore agency to its true location in the minimal living units, the cells and the aggregates of cells. So Dennis Walsh has put it this way. He said that genes collectively, and I'd actually say genomes, have it in their repertoires to produce the appropriate output under previously unexperienced circumstances. Now, in the post-genomic era, molecular biology has then become a study not so much of what life's molecular components are, but of how they're deployed and used in different circumstances. And so attention has shifted increasingly from a characterization of the protein coding genes and their gene products to the investigation of gene regulation. And in general, agential behavior is linked to regulatory mechanisms because it demands a flexible and contingent response to circumstances at all levels of the organism, from the genome to the whole organism, using the same set of molecular resources in new ways. And gene regulation, therefore, I think can be considered to lie at the core of agential behavior. Now, when it was first encountered in the 1960s by the work of Monod and Francois Jacob. It seemed to have a kind of digital algorithmic logic that very much fitted with the computational paradigm of the time. So, in that view, one gene could, via its gene product, switch another gene on or off, as in the classic lac operon, where the lac repressive protein is constitutively bound to a promoter site of lac genes unless it's liberated by binding to lactose. But for a multicellular, multi-tissue organism like us, a rather different sort of logic seems to be required. To make such an organism, when all the cells draw on the same set of genetic resources, there need to be ways of using different suites of genes in different cells, both to shape development and in the moment-to-moment responses of cells to the circumstances that they have in the mature organism. This has long been recognized, of course. It's why Conrad Waddington coined the term epigenetics in the 1940s to describe the differences in phenotype or behavior that don't derive from differences in genotype. And many of the basic molecular mechanisms of epigenetics are well known now: the methylation of DNA, the modifications of histones. What's far less clear is the logic, and some people like to call it a code. I'm not sure that it necessarily will prove to be that, but the logic of these epigenetic marks. It's certainly not as simple as saying that, say, a methylation mark on DNA inhibits transcription at that point. But a significant part of gene regulation is not about these chemical changes to chromatin, these sort of overwriting, if you like, of chromatin, but about the process of how transcription to mRNA and translation to proteins, how that is controlled. And I want to say a little bit about how that works for metazoans like us, though I should start by making clear that there's still an awful lot about it that we don't understand. So as with the lac operon of E. coli, whether or not a gene gets transcribed is partly governed by these proteins called transcription factors that bind to DNA and affect recruitment of the RNA polymerase to that chromosomal region. And as with the lac operon, this involves promoter sequences near the gene being regulated where the transcription factors bind. Okay, but beyond those similarities, the situation is very different in eukaryotes and in complex eukaryotes in particular. For one thing, in general, our transcription factors don't seem to show on their own strong selectivity for how they bind DNA. Our genomes have sequences all over the place that could bind a given transcription factor. And our transcription factors tend to work in pairs or in groups. They work combinatorially and cooperatively. And again, while some people like to talk about a second genetic code governing these binding patterns, it's far from clear that there really is going to be something like that, a code worthy of the name describing this. For one thing, the action of our transcription factors is typically highly dependent on context. So the same transcription factors might bind to different regions of DNA and different cell types, and a given regulatory sequence next to a gene might be activated or inhibited by different transcription factors in different cell types. Most transcription factors aren't simply repressive or activating. They can show either function in different contexts. And in fact, there's relatively little correlation between whether a given transcription factor binds at a regulatory site and whether expression of the corresponding gene actually changes. There are some instances where you can deplete a transcription factor in a cell and you see a change in expression factor, even though there is no detectable binding of that factor to a promoter site at all. So it's really hard to discern what the general rules are to this complex behavior, other than to recognize that they're going to be fuzzy rules. They have none of the digital precision of at least some transcription factors in prokaryotes. But surely part of the reason for this puzzling diversity and complexity of transcription factor effects on expression is that transcription factors aren't the only players involved in eukaryotic gene regulation. There are typically all these other elements involved too: cofactors, non-coding RNAs, other regulatory DNA sequences. In particular, gene regulation is influenced by these sequences called enhancers, and these are often situated not close to the gene, but a long distance away, maybe a few 100,000 base pairs away on the chromosome. it seems that these enhancers are brought close to the regulatory site to interact with all these other components by being extruded on huge loops of chromatin. And this is a very controlled, orchestrated process in which the loops seem to be constricted at their base, typically by the action of these two proteins, CTCF and cohesin, and then are pulled through that constriction site by motor proteins. Now, at first glance, this looks like an absurdly complicated way for gene regulation to happen. Why on earth would evolution do this? Why would it place a regulatory region, for example, so far away and then have to go to all this effort of pulling it close to do its work? One possible answer for that is that this allows, again, for more combinatorial complexity in regulation than would be possible if you were trying to pack all of these components next to each other. It also means that a given enhancer can influence the expression of more than one gene, because in principle it can be pulled over to different locations, again, creating more combinatorial options. So the idea then is that the appearance of chromatin loops and of these distal enhancers created possibilities for doing much more with more or less the same set of genes than was feasible when regulation was still linear and proximal. And indeed, that's what the evolutionary record seems to bear out. Besides these loops, there's a higher level, or there are several higher levels of organization in chromatin that influence regulation and the co-expression of genes. Except during the onset of cell division, chromatin exists, not in those nice little. Sorry, that's the work I wanted to talk about, the evolutionary aspect of the development of these loops. So yeah, this is really what chromatin tends to look like most often. It's it's a tangled mess. And it's partitioned into dense regions called heterochromatin and more open regions called euchromatin. And it's generally only the latter, these more open regions, that are accessible to the transcription machinery. Chromatin seems also to be divided into compartments, sometimes called topologically associating domains, or TADs, within which the genes seem to be co-regulated and co-expressed to some degree. This organization seems to be important for defining different cell types and different tissues, but really, I don't think it's well understood at all at the moment. My key point is that regulation also depends then on these higher order three-dimensional features of chromatin that are responsive to their environment. So once the complexity of eukaryotic gene regulation began to be apparent in the early 2000s, to begin with, researchers tried to fit it into this paradigm of the paradigm of the time of precise molecular recognition and transfer of information by arguing that somehow all these different components assemble into some precise stoichiometric complex. That sort of thing, when you think about it, made no chemical sense whatsoever. The idea that you could assemble such a structure by diffusion and then expect it to hold together with everything in place while all the elements are sort of unbinding and binding was just a fantasy. What seems to happen instead is that all these regulatory components are gathered into rather loose molecular aggregates that are less like machines and more like committees that reach some sort of collective decision. These aggregates seem to be a genuinely different phase from the surrounding fluid of the cell nucleus, and they're often regarded as examples of what are called biomolecular condensates. And these condensates hold together all the various components in this loose ephemeral assembly via weak and relatively non-specific interactions that prevent them from being dispersed by diffusion while they do their job, acting as a kind of a membraneless compartment. And it's now recognized that, in fact, biomolecular condensates do all sorts of jobs in the cell. Since they were first identified in 2009, they've been found to be involved in things like protecting cells against stress. That's a particularly important role, repairing DNA, in sequestering and storing molecules, in the creation of functional catalytic chemical and pH gradients, and all sorts of other roles. They're typically composed of proteins and or non-coding RNA molecules. So they seem to represent a previously neglected level of organization in the cell, with a strong sensitivity to environmental signals such as temperature or stress. Some pathologies are linked to the question of whether or not condensates form, and it's a question that can't be meaningfully reduced to looking at the sequences of their component parts. They're too loosely specified for that. By aggregating many diverse components in a context-dependent way, condensates can act as sort of integrators of contextual information, which I think is the hallmark of agency. And in this recent work by Yves Barral and his co-workers in Paris, they've described such a condensate-mediated process of kind of decision-making in budding yeast. So yeast cells have a range of behavioral options depending on their circumstances. They can proliferate, they can have an active cell cycle, or if nutrients are scarce, then they may enter a quiescent phase. If there are potential mating partners nearby, then pheromone signals can trigger the cells to differentiate into a mating competent state. If that happens, but then the cell fails to find a mating partner, then it returns to the proliferating state. So there's all sorts of decisions these cells have to make. And those decisions, to make it even harder, those decisions seem to be influenced by the age of the cells. The decisions seem to be processed by the formation of condensates made from an RNA binding protein called, I don't know how this is normally pronounced, it's WHI3. I'm going to call it WE3, which regulates, this actually regulates the expression of a cell cycle protein, a cyclin. So WE3 condensates interact with another type of condensate, a stress-related condensate called P bodies, which are more likely to form as cells get older and which may suppress a response to the pheromone signals. So it makes older cells less likely to mate. So somehow then this combination of WE3 condensates and P bodies supplies a system for integrating all this contextual information about age and about mating opportunities to allow the cells to arrive at an appropriate fate decision. So this sort of behavior, I think, might be best interpreted not in algorithmic terms as a kind of decision tree, but by analogy with cognition. It contains the elements associated with, I think, a minimal definition of agency. None of this then follows the standard biological paradigm in which molecules have precisely defined structures and functions programmed via sequence information that's highly conserved and is highly sensitive to mutations. There are different molecular design principles at work. The fuzziness of these interaction strategies seen, for example, in eukaryotic transcription factors reflects the fact that those molecules belong to a large class of proteins said to have intrinsically disordered structures. So rather than having these well-defined folded structures like hemoglobin here on the left, there are substantial parts of these proteins in which the peptide chains don't really fold at all. They have rather labile and irregular conformations. And it's estimated that around 30 to 50% of all our proteins are intrinsically disordered to some degree. But we've only come to recognize this over the past 2 or 3 decades. So the old paradigm, the kind of central dogma paradigm really, in which protein sequence uniquely determines structure and therefore function, that paradigm no longer holds in general. Disordered proteins tend to be more promiscuous and less selective in their binding. Some of them actually fold on binding, but some of them stay unstructured when they bind. Their specificity, to the extent that they show it at all, tends to come from quite general chemical properties like hydrophobicity or charge, rather than from highly sequence-dependent secondary structure. And this fuzziness is clearly by design, as it were. The amount of disorder in the proteome of an organism increases with organismal complexity. So it suggests that evolution has found this to be a useful principle for orchestrating complexity. Transcription factors themselves are often seen to be paradigms, really, of intrinsically disordered proteins. Here's an example of one, and it shows that they're typically this combination of well-ordered domains and of disordered domains. And the promiscuity of interaction that the disordered regions confer seems to be an important aspect of gene regulation. And it's surely no coincidence, too, that condensate forming proteins are typically disordered, given that condensates typically involve these rather weak, loose, low specificity, low affinity intermolecular interactions. Intrinsically disordered proteins have just the right properties for forming these sort of gel-like networks of condensates. So, in short, disorder offers cells ways of feeding contextual information back into the structures and functions of the molecules themselves. Some of that contextual information is also integrated by this complex, the spliceosome, and also by other post-translational regulatory processes. So the spliceosome is a multi-component system. It's controlled by splicing factor proteins that belong to a large family of proteins called RNA binding proteins. And the spliceosome also makes use of small non-coding RNA molecules, which can vary in different contexts. So we might think of it too as a kind of contextual integrator of information. And I want just to turn briefly now to these non-coding RNAs, which represent another way that the molecular paradigm has shifted in post-genomic biology. It's become clear in the past 2 or 3 decades that a considerable amount of the non-coding genome, that is, the sequences that don't encode proteins, a considerable amount of it is used to produce functional non-coding RNAs. And we seem to be finding more and more families of these non-coding RNAs, many of which are regulatory. So many small regulatory non-coding RNAs, including microRNAs, and there's an analogous class so-called Pee-wee interacting RNAs that seem to function as a sort of molecular immune system against transposable elements inside the cell nucleus. Many of these small non-coding RNAs work combinatorially. They're just too short to be able to carry much functional information. Some of them are, barely 24 base pairs long. And they seem to be, again, non-specific because of that. So a given microRNA may target many different mRNAs in the way that they regulate that process, that post-transcriptional regulation. And any particular mRNA might be targeted by many different microRNAs. There are other longer non-coding RNAs that seem to function as structural elements. For example, some of them seem to prop open regions of chromatin so that they remain transcriptionally accessible. Some non-coding RNAs provide the scaffolding for biomolecular condensates, where again, their functionality seems to depend much more on their chemical nature as charged polymers, say, rather than on any highly specific sequence information. And some people estimate that there are now more non-coding genes known than coding genes in the genome. I should stress that these numbers are somewhat controversial because it's very hard to assign function in general to non-coding RNA transcripts, because many of them are expressed only at very low levels, and some of them are in fact likely to be just transcriptional noise. But some of these non-coding molecules do their job only in very specific cell types or at a very particular developmental stage. So it's very easy to miss the time or the place when they are actually functional. I think the fact is that actually non-coding RNAs challenge traditional distinctions between what is functional and what is not, what is just noise at the molecular level. Some of them seem able to be produced as what we might call potentially functional matter, able to assist some molecular processes in a kind of speculative manner. And this ambiguity seems to apply also to genomic matter more generally. It appears to be an important feature of the genomes of complex organisms, that they can develop redundancies and duplications and adventitious additions that seem to sort of provide a pool of material that might be capable of developing functional roles in the future or in particular circumstances. And in this respect, I think what's regarded as noise or junk and what's regarded as functional is not necessarily as clearly distinguished as we once thought. Cells seem to be actively engaged in the generation of possibilities and contingencies at the molecular level. So one emerging theme here then is that many functional biomolecules operate collectively and combinatorially, such that we can't meaningfully assign the individual molecules particular biological functions at all. So it's a kind of causal emergence, and we might regard this as a way that biology avoids baking in decisions and functionality at too early a stage. Decisions are made in biology, of course, at many levels during development, for example, in a manner that might best be considered as a kind of a mobilization of the agency that the individual cells have. So this really is a kind of summary of what I think of as the some of the key aspects of the new molecular biology. And I'd just like to point out finally that they take us back to an old debate. In the late 19th century, the demise of vitalism led a number of physiologists, really, then, particularly Eduard Pflüger in Germany and Jacques Loeb in the US, to promote a mechanistic model of biology in which life could and should be understood in a reductionistic and mechanical way. And this perspective, of course, rose to dominance with the inception of modern molecular biology in the 1950s. Set against that view was a position proposed by, among others, William Ritter in the US, the founder of the Scripps Institute, and J.B.S. Haldane in Britain, who argued that explanations of life couldn't afford to lose sight of the whole organism. So this is the view that became known as organicism. In much the same spirit, the hegemony of genetic and molecular explanations in biology was challenged in the later 20th century by Waddington and others, who felt that their discipline of embryology, as it then was, or developmental biology, as we'd say now, in which higher level processes were given equal attention as genetic mechanisms were, they felt that this perspective was becoming eclipsed. Today, I think something like an organicist perspective is returning with an advocacy for focusing on the cell or on processes of self-organization or self-regulation or tissue mechanics and physiological feedbacks in controlling biological development and behavior. And these two viewpoints, the reductionist and the organicist viewpoint, are commonly presented as antagonistic, as sort of battling it out, as mutually exclusive, really. An examination of the molecular mechanisms through which goal-directed yet fluid features of agential behavior can arise, I think reveals that that opposition is unnecessary. The molecular characteristics of complex organisms seem to have been tailored by evolution precisely to convey contextual and evolvable outcomes that are robust against molecular stochasticity. These mechanisms aren't machine-like in any sense that compares with human-made machines. They don't depend on a precise specification of parts or processes, nor do they enact any encoded plan. They achieve robustness not by simple redundancy, but by loose specification of higher order principles and multi-level causation. I think we lack an effective metaphorical language to describe this mode of operation, which is very different from any so far used in technology. So I'm very interested in exploring with a group like this what form that new language and those new narratives might take. So that's all I wanted to say. Thank you.
Thank you so much, Philip. That was amazing. So well organized. Um, Fantastic. So guys, what we're going to do now, I'm going to ask a few questions of Philip just to sort of anchor the discourse. I've got four questions here that I've came up with before this meeting, but now they feel like they're even better. And then we're going to go into, open discussion until 11:30, so another hour and a half almost. And you guys keep in mind a couple things. One is that I mute you when you're not talking. So if you have a question, raise your hand and get in the queue, and then I'll call on people. And I've been warned that I need to sometimes be a more aggressive moderator. I tend to love it when people get into a long back and forth, but try not to do that too much. So if you ask a question or make a comment, try to limit your follow up so that we can move through and people can speak. So. Um, yeah. So anyway, thanks so much, Philip. That was amazing, and I think you've opened up most of the Pandora's box of implications that we love to get into. Um, the first one, the thing that, and I'm so glad you said the thing about the metaphors of biology, because we just have a really hard time getting away from our machine-like metaphors. Um, so if I'm understanding what your presentation just now, and just the general thrust of your work is that for some reason, we have all this digital precision at the lowest level. We have this precisely error-corrected code, or it appears to be a code. It's an error-corrected sequence that's like corrected by physiology even more than it is by self-replication. So, and of course, the accuracy of the code depends on physiology, a physiological process. So we've got all this digital precision at the lowest level, but then it completely disappears at the level of gene expression and physiology. And so that to me is this huge mystery. Like if you're going to be very precise, why waste it when you get to these higher levels? And in your book, so How Life Works, on page 341, in case anyone's tracking this, you've got this great metaphor. You talk about dust devils, and you say, like, we are dust devils, sort of, but dust devils are spun up afresh, which is a great phrase, spun up afresh, and they don't inherit anything. So I feel like there are these two oppositional concepts. One is like there's some sort of spontaneous ordering process that's spinning up an organism. On the other hand, there's this tremendous fidelity to this inherited sequence or inherited something. And what is the relationship between those two things? And you had just proposed 10 pages earlier this thing that really stuck in my mind, which was about Lindquist and these Drosophila genes that are they call them morphological capacitors. And you say something like, in a way, the sequence of the gene here is acting to suppress variation, to suppress possible morphological outcomes. And so I just wondered if you could kind of comment or go with that for a second. The idea that, the dust devil part might be something that's actually causing the plasticity. And then the ordered part is something that's, causing the fidelity, and how those two might relate. Or because the conventional view, just to contrast, the conventional view is that the ordered part is responsible for both the plasticity and the fidelity. Um, yeah.
Yeah. Um, okay, quite a number of things to say about that. I think, first of all, the way I'd think about it is that it seems, unquestionable that for complex organisms, again, in particular, a lot relies on processes of self-organization and, things like what happens at the tissue level, the way it folds, the kind of potentiality that the materials themselves have. I mean, Stuart Newman has talked about this, and I think about it as the genetic information is steering those processes. It's not creating them in any sense. They come for free, if you like, and the genes are steering those, and in some ways literally shaping those. So the two are coexisting and supporting one another. And in that process, the information, the precise information at the genetic level, sometimes, of course, it really does matter. There are plenty of proteins, plenty of enzymes that really do have precise, well-folded, ordered shapes that they need in order to do their task. And a lot of these are really kind of, housekeeping enzymes that you really have to have and that are highly conserved in evolution. So that's clearly important. And a lot of those, I mean, they're highly conserved and also often evolutionarily ancient. So they're, all organisms need to have them. So all of that stands to reason. And, that precision seems to be particularly important in prokaryotes. So we have to have that. But I think that it's been I think it is overstated that, we sort of get used to, well, of then, all of that information matters. And we see more and more that some of that information really doesn't. And this has been one of the big sort of discussions about, for example, whether these non-coding RNAs are truly functional or not. There's a camp of people who say, well, they can't be because they're not highly conserved. And functionality is considered to be something, if almost by definition it has to be highly conserved. But when we understand how those molecules and how disordered proteins do what they do, we can see that there's no there's no strong pressure for them to be highly conserved, and they are not. That there's a lot of variability in them, because what really matters is more sort of generic properties. You can probably substitute one hydrophobic residue for another without anything becoming problematic about it. So I think the notion that everything, all the important information is highly conserved is one that seems to be increasingly unclear, and increasingly under challenge. And we can understand why that should be so when we understand the way these molecules are functioning combinatorially and through these kind of loose, fuzzy interactions. So I think, it makes sense that both of them are there. The work you mentioned by Susan Lindquist, so it's looking at these heat shock proteins, which are they're kind of chaperones. They help proteins to sort of fold properly. But what they also seem to do, I'm really interested in looking at them at the moment as examples of systems that enable variation to happen but to stay hidden. This is I've been speaking to people about this recently, and I think it certainly feels clear to me that we don't quite understand. There's this delicate balance that seems to go on in nature, and particularly again in complex organisms, where variation, rather than variation being the stuff that evolution is working with, a lot of variation is just kind of hidden and compensated for at higher levels, and so it just builds up. And that's really what this sort of evolutionary capacitor seemed to be about, that these proteins are part of the system that allows that variation to build up without being deleterious. And then there comes a threshold where you've just got so much of it, something's got to give, and you suddenly get a really big transition to maybe a completely new morphology, completely new possibilities for body shape or whatever, or for something catastrophic. So it's not, the view seems to be that evolution isn't this steady, gradual accumulation of small changes, that it's actually this combination, probably quite ad hoc combination of the system is set up to be robust against variation, because otherwise how could it evolve at all? But it can only do that for so long, and then suddenly you get evolutionary leaps. And that too seems to me to be tied up with the looseness of specification of parts, that that too allows variation to happen, which of course it will happen without it being catastrophic. So all of this is about how to make really complex systems that are evolvable, and it seems pretty obvious to me that you will not get evolvable complex systems if everything matters, if all the information has to be passed on and has to be enacted in a very precise form. You have to have this looseness and flexibility.
Yeah, that's awesome. Yeah, so the hidden variation is really fascinating because, like you said, there's this like smoothing process at the higher level. And so you gave a great example of it when I think it was, oh, I'll clear the page, but 456, but you're talking about Darwin's finches, and you make the comment that, like, these gradual changes in beak size, um, you know, are are much more complex than they seem to be, right? Because it's not only that the lower beak changes, the upper beak changes at the same time, and it's not only that, but the shape of the head changes also, and the blood vessels and the innervation and everything changes all together, and it's sort of smoothed out. And in a way, those those could be considered as independent changes, and yet they are smoothed together to happen all at once. And so yeah, like why are they smooth? Like why, like why is it that why is it like that? I mean, why does development smooth out inconsistencies rather than amplifying them or, you know,
Yeah, well, th this is the idea that Mark Kirshner and John Gerhardt have been, working on for some time in thinking about evolvability and in thinking about how it can be that organisms somehow are able, in the face of mutation, to still, if you like, deliver viability. Um, and it does seem to be that somehow there are these higher level processes that work with, whatever comes lower down in a way that integrates it all together. And I was really interested to see that they too had concluded that if this is going to happen, fuzzy molecular interactions rather than precisely specified ones are precisely what you're going to need to have that kind of that slack, I suppose, in the in the lower level system that can still be put together to make a viable organism. So I'm really interested in this in this ability of nature to do things this way. I don't think it's well understood. I certainly don't understand it well enough myself, but I think it's an absolutely key feature of how evolution happens. That again, that too is not all bottom up. In fact, it has to be somehow robust against bottom up variation.
right. Yeah. So I have to somehow suppress it. Um, so your epilogue in your book, it has this long essay basically in a box. That's one of the best essays I've ever read about biological agency by far. Um, and so one of the things you're asking, so you talk about the extended evolutionary synthesis. And so we're kind of, in some ways, all of us here are sort of an outgrowth of that. And you talk about, you know, Leyland and colleagues, they argue that, okay, there's also these top-down processes where organisms act on their own, to cause their own sources of variation in various ways. And, of course, it's a circular process, so it makes sense to you. I think you're saying that, effects become causes, right? But you ask the question, one might even ask whether evolution causes organisms themselves, or rather, whether it is made possible at all because organisms are the way they are. And so, I just wanted to like kind of push you to extend that idea a little bit. And like, I guess my provocative way of asking the question is are organisms holograms?
Right. That's an interesting way of looking at it. Well, I'll start, Charlie, by confessing that that box at the end of the book is there at the end of the book because of my timidity, you could say, about delving into evolutionary questions. I realized, everything I say in that book um, speaks to even aspects of evolution in a way that I don't feel I understand well enough, except to know that I know enough about it to know that these questions are incredibly subtle, and that to do justice to what these molecular and cell scale emerging ideas mean at the evolutionary scale would require another book, and certainly one that I'm not qualified to write. So that final box was my kind of attempt to say, okay, I realize there are these unanswered questions from the evolutionary point of view. I'm just going to put this out there, and I'm actually I come from that question actually quite conservatively, to be honest, simply out of ignorance. I feel like it's tempting to jump in and say this changes everything for evolution, and I'm nervous about doing that because I don't think I understand it well enough. But nevertheless, it seems clear to me that once we recognize genuine agency in living things, that has not been there in traditional evolutionary theory. I know that there's all the discussion about whether niche construction takes care of that. I was very interested to find out, quite recently that evolutionary biologists also talk unproblematically about what they call behavioral drive, which is basically the organism's doing new stuff because it wants to do new stuff, and then it leads to evolutionary consequences. It's basically, agency affecting the course of evolution. It seems very, very clear. It's all, so it's always been there, and so this reluctance to speak the word agency in some evolutionary circles seems bizarre because it's absolutely in there, in at least in this concept of behavioral drive and probably also in niche construction. So these questions are there. To my mind, the way where I feel I have some kind of foothold in those questions is about this issue of evolvability, because that really seems quite directly to link back to the molecular scale processes that are going on and how you can make a system like this that is incredibly complex and yet not extremely fragile against small changes. That to me seems one of the central questions about the evolutionary process, however it happens. And it's absolutely not clear to me that a kind of strict, gene-centric view of how organisms work is going to explain how that could even be possible. So that's my way in, but I'm afraid I'm going to kind of duck it and leave it to you guys to try to figure out what, if anything, this level of thinking about agency at the level of molecular interactions, what it might mean for the evolutionary process and whether it means that we have to do some profound rethinking or only some tinkering around the edges. That's really the question that I sort of leave open in that final part of the book.
Totally. Yeah. Totally. And yeah, the question of agency and the question, it raises the question of teleology. And that, as you point out, it's sort of inescapable question of teleology. So my last question is this. So um, a lot of thinking, even totally up to date thinking about these issues, leaves in place the teleology of survival and reproduction, basically. It leaves in place the assumption that, um, you know, why this all makes sense is because it's all trying to get itself into the future. But as long as we leave that assumption in place, I would say we also leave in place the erasure of the organism itself. And we're talking now in terms of a lineage, and it's the story of the lineage. And it always drives us back to what is it that's inherited, right? And it drives us back to the question of, DNA, to the centrality of DNA, if we leave that assumption in place. And so one of the things tried to do in this group, we've explored a couple of alternate teleologies. And so one of them is the idea that Richard Watson and I have been developing called epistolution, which is the idea that organisms are trying to understand the world. Like the dust devil part is a drive to, or a physical process that's trying to accumulate, a causal sense of what physical forces and entities are important in the world around. The other one we've considered is Mike Levin's idea of the ingression of Platonic forms. And it's a pretty wild concept, but you can look into what he's doing there because it's really fascinating. But I just wondered, in all of your explorations, have you encountered or thought of any alternative teleologies or like um, fundamental teleologies out there?
Um, I I mean, of course, the whole question of of admitting teleology was one that Ernst Mayr tried to deal with by coining this term of teleonomy, and I think I agree with what Alan Love has said about that it's really that doesn't add anything. That's a way of avoiding the taboo word, but it's basically the same idea. So why not admit that organisms themselves, are teleological and they have purpose. They are trying to do things. And I mean, I had actually just earlier this week, I had a fantastic discussion with Dan Nicholson, about that, about these issues. And, Dan pointed out, put it very elegantly in sort of saying the gene-centered view is one that, of course, famously has no need of the organism, that it looks at, it places all causality at levels below at the genetic level and levels above at the population level. Those are the two elements that it engages with, and organisms disappear. And it just feels to me as though, that that cannot be right. Organisms are the things that evolution sees, if you like. So, there has to be something missing from that picture. But I guess the book, my book was very purposely called How Life Works rather than How Evolution Works, or rather than What is Life, which, of which there are several books. Because I was interested in this question of how organisms work, what are organisms, taken at face value. And for me, the evolutionary process is, it seems to me, I have the luxury of regarding it simply as the process by which these goal-driven, these purposive entities are produced. That is what evolution does. It produces things with purpose. And in the course of that, I say in the book, in the course of that, it also produces a sense of meaning, that things mean something to the organism, that the organism is involved constantly in making value judgments. How much should I pay attention to this stimulus as opposed to this one? How much can I afford to ignore? What information from this overwhelming amount that the environment is giving me is important? It means something to me, has value to me, and what does not? And I absolutely think that we need to understand organisms that way in terms of meaning, makers of meaning and value. So for me, evolution is the mechanism by which such things are generated, and it's the only, at the moment, I feel it's the only mechanism we know of by which things of that nature can be generated. So again, the question of how that feeds back to the process of evolution itself is a really difficult one that I'm glad people like you and Richard are thrashing out. And I'm taking the kind of the luxury of just saying, well, I want to look at what it gives us, what it produces.
Totally. Great. Great. We have some questions in the queue. Henry, Cy, Joanna, then Kate. So, Henry, go ahead.
Hi, really enjoy your talk.
Thank
So
you, Henry.
I have some question you are just discussed is because I think many of the question you addressed and also some of the confusion molecular biologists face actually is about what is the mechanism of evolution, right? So you mentioned this a couple of places, but the key we realized if we use the information perspective, we actually have better understanding of the evolution. Because no matter which successful species, they will be replaced. Why? Right? So give people the impression, in fact, that reflect the process for the information increase complexity and diversity. So if in that way, we thinking organism just a carrier for information. So therefore, all the fuzziness, all the jumping and gradually evolve of the gene makes sense. So we put the two-phase evolution, right? First phase, the macroevolution, you create a new system by reshuffling the genome. You do not need to change the transcription factor. You do not need to change, but you change the information context, right? That's what you have talking about in such a way. But after that, evolution is all about the conservation. It's not the innovation. Because the conservation to duplicate this successful unit, grow up the population until the crisis kicked in. they reorganize again through the chaotic process. We're talking about the genome chaos. So the further inheritance is one of the key features for this system, because to answer the Charlie's question, the gene coding is not the points of phenotype, it's the spectrum of phenotype. And the environment actually makes the setting of selection. So in this case, that is pretty much like the condensing everything, right? So they do not need a specificity, because specificity is a feature of physics. So when you build up, the system become more complex. The fuzziness become constant increase. But they have different level of fuzziness. They have genome level fuzziness, gene level, epigenetic level. Also, there's some pure chemical level. Because for biological, we always thinking specificity is we always use one pure enzyme to do the one substrate to do the experiment. But if you put a 1,000 enzyme into the test tube, you immediately know all the logic in biology we know is flawed, right? So they do not give you specificity as such. So therefore, the specificity is driven by the time and the care by create the context. That's all the biology is create the context. So now we're thinking the separate biology. I need to ask you advice on this one. Is the information capability, because biology, the first is information self-creation. So all the physics systems don't have such a capability. So the self-creation, especially at the species level, is fundamentally easy. They just reshuffle the chromosome every time they become new species. And after that, they just need everything to preserve the information. So that's why the gene is still very important. Genome is still fundamentally important, because without those, you have a miracle, you have emerged a miracle. But this emergency and this self-organization cannot sustain. So therefore, you need the mechanism to preserve such. So therefore, we're thinking the information self-creation may be equally important as the self-organization process. But the biology beautifully puts them together, so they make the miracle become biological routine. So when it becomes biological routine, it's no big deal. They just come precisely doing that. So that's the idea we actually try to push it. I just stop here, but I need ask your impression on this.
Yeah,
Yes.
thank you, Henry. Yeah, it makes me think. I very recently had a discussion also with Chris Adami, the information theorist at Michigan State, who I think voiced probably a similar point of view to yours, in that his view of evolution was an informational one, and that in his view it was simply about the perpetuation of information. And in that sense, again, organisms just become the vehicle by which that can happen. I have to say, to me, that always sounds, well, for one thing, it always sounds like it's close to committing the biological heresy of attributing teleology to evolution itself. That the purpose is almost like the purpose of evolution is to perpetuate information, and there are reasons to feel uneasy about that sort of attribution of any kind of purpose to evolution itself. To me, the purpose resides in the organisms rather than in the evolutionary process. The evolutionary process is simply what gives rise to those. I'm trying to figure out whether it's just a question of perspective or whether it's something more than that, because it feels to me as though seeing evolution that way has the sense that somehow you're wanting to sneak out the agency from where it belongs in the actual agents themselves, the organisms, and put it into the evolutionary process. I'm not saying you're doing this, Henry, but I'm saying that's the danger that might happen, that the agency, it's always going to be there in every biological narrative. It's there, and if we don't put it in the right place, it's going to end up in the wrong place, and that's the concern. I also wonder, though, about the question of this notion of seeing evolution as information wanting to perpetuate itself, if we put it that way, because again, exactly how does that operate? Because again, there are some sequences, some genes that are highly conserved, and there are sequences that are not. And there is, an interaction between the two. I mean, if we just think in terms of, say, transposons, there it's almost like that's like pure chemical information trying to perpetuate itself. That makes a lot of sense. And what that does is that it creates havoc in the bigger informational system of the genome, which doesn't get perpetuated, in quite the same way. So I'm not clear about in what sense we could think of evolution in general as being the perpetuation of information, rather than there are aspects of the way that that process works in which information does get and does need to be conserved. And there are other aspects where it really doesn't matter, because what is happening is a higher level process that does not require on precise specification at the genetic or at the molecular level. So I don't think that will answer your question, but I think maybe it sort of complicates some of the issues that you were talking about.
Great. Great. Awesome. Thank you. Sai?
Yeah, I really enjoyed your talk, and I also love your book. My question is simply about gene regulatory networks, which you didn't mention, but I'm wondering if that adds another level of complexity to the whole question of gene regulation, because these networks, you know, basically regulate themselves as well as the expression of other genes. So, and in fact, in eukaryotes, the complexity of those networks is really mind-boggling. So how does that fit into your model?
Yeah, that's another level. So, I talked about the molecular level, I talked about condensates a bit, but gene regulatory networks are absolutely another level of the hierarchy in which a lot of the same considerations apply. That it seems, that there too, I feel that rather than seeing them as somehow, I mean, gosh, Stu, I'm going to put my foot in it here, but rather than seeing them as Boolean networks in the sense of, simple digital ones and zeros doing stuff, I would tend to want to think of them more as being like. Oh, I'm sorry, I've got an echo here. I wonder if someone.
500 jobs they promised to stimulate the economy here.
Is it?
So really what we're talking about.
I'm getting a bit echo now. I don't know if anyone wants to hear that. I don't know.
Because of however the
I'm
hear a voice.
Sorry.
decisions that
I figured it out. There we go.
Brilliant. Thank you.
Sorry,
It's
Simon. Sorry about that.
Yeah, I like to think of them almost as having that sort of cognitive character, rather than that sort of digital, microchip character, in that they do seem to have this adaptability. And I think the thing that I would say about that side, from the point of view of what I was talking about, is that I think it is absolutely no coincidence that very often the hub molecules, the ones that connect many different parts of the gene regulatory network, are disordered proteins, are proteins that are able to form interactions with a wide variety of substrates, and again, that do so, I think, in this adventitious way, that do so in terms of having the potentiality to form new connections within the network, and that is essential to the way they function, that they're not precisely digitally defined. They seem to have these hubs that can at any moment, forge new connections that might or might not be useful. And not just useful in evolutionary terms, but useful in the moment, useful in proximal terms. And they seem to have that character, that a lot of the interactions that might be involved in those networks are ones that might turn out to have no useful function, and so those fall by the wayside, but they have the potential to form new ones. So I think that gene regulatory networks provide a very good example of why it is that these molecular principles of promiscuity and relatively weak interactions seems to be one that evolution has found to be extremely useful. The more complex you need your regulatory network to be, the more you need these these low specificity, these promiscuous molecules, and that's precisely what we seem to see.
Thanks. Thank you. Joanna?
Hi, everyone. Hi, Philip. It's great to
Hi.
meet you here. I have chased you online a bit in the past, but
We have got to
finally
meet
we
up,
meet.
Joanna. Yeah.
Finally we meet.
Yeah.
So my question, I would love to nerd out about how there's a lot of redundancy in prokaryotes and even in the prebiotic world where I work. But given that I have you here, I think I really want to touch the elephant in the room, which is why hasn't the paradigm changed? There's enough science, there is enough demonstration of complexity of emergence all over. Most of us scientists don't like Dawkins anymore. However, everywhere else, people are still with the selfish gene narrative. You talked about something which I think is very important in the end of the talk, which is we don't have the right metaphor. Many years ago, I thought Dennis Noble's Music of Life, uh, the dancing of life, the orchestra of life would be good ones. Apparently, they weren't good enough. Can you talk a bit more about that and why we haven't been able to popularize this new paradigm of biology?
Hmm. Great question, Joanna. I think it's a complex answer. As it happens, I have just published, went online yesterday, an essay touching on this question and looking at the wider question of narratives in science, and how I think it's underappreciated what role they play. We're quite the history and philosophy of science is perfectly comfortable talking about theories and theory formation and testing theories in a Popperian way, and all scientists kind of know this is what they're meant to do. The one thing that has struck me over years and years of writing about science is how strongly scientists cling to their narratives, much more strongly than their theories. They're reluctant to let theories go because we're all human, but they know you're kind of meant to, and you're kind of meant to test and make experiment. But my goodness, narratives just stay there, even if they have to slightly bend or even severely bend the narrative in order to preserve it in the face of new experimental results. That is what they do. Because that is because narratives are how we frame our thinking. And if you abandon a narrative, you've got to really rethink, quite fundamentally in a way that a theory doesn't require you to do. So I think we need more attention actually given to the question of how narratives develop in science and how they determine agendas, research agendas. And of course, there are sort of obvious vested interests in certain narratives. The Human Genome Project was predicated on a particular narrative about what the genome is. That is still the narrative that is on the institute. I forget the full name that looks after that side of things at the NIH. And it's still a strong narrative that comes out of genomics more generally and is giving rise to really quite worrying concerns now about worrying trends now about a resurgence of genetic determinism and eugenics. So that was what the human genome was predicated on. The fact that I think partly as a result of having done that work, we realized actually this isn't where it's not just that we haven't yet got to the answers or that things are actually more complicated than that. It was the fact that it showed us we have the wrong narrative. The idea that we went into the Human Genome Project thinking we're going to look at all the, it's all the protein coding genes that matter. And we've come out of it finding that there are so many different regulatory regions and so many non-coding genes and, a fuzziness between the two. The whole genome looks different to what we thought it was. And yet somehow that same narrative is still there. But, that's so in that case, a big, expensive, high profile international project was predicated on it, and a lot of careers went along with that. And I think what it gives rise to, I think the reason perhaps it's persisted so long in biology is that there's always another ohm that you can go to. You can always say, you can, that, okay, well, no, we need the proteome. Oh, we need the metabolome. Oh, we need the interactome. Once we have that, we will be able to answer these questions that we couldn't have before, rather than thinking, perhaps we need new conceptual thinking rather than just more data. And I think people are starting to realize that. But I guess it's also because many of these things, I felt like I was saying again and again in the past 2 or 3 decades, because that's what seems to have happened. There was really no talk of non-coding RNA, before. I mean, it sort of started in the 90s with Rufkin and Ambros's work, but it only really took off at the turn of the millennium. The same with disordered proteins, which weren't studied before, not because they weren't there and they weren't important, but because it all depended, or mostly depended, on crystallography, which isn't going to give you results for disordered proteins that you can't crystallize. So I think that a lot of these new narrative changing discoveries in molecular and cell biology have only come about in the past 2 or 3 decades. And so perhaps you could say it's still, relatively early days. And I do see some sign that the narrative is changing, or a willingness to think about, or have to think about, new ways of understanding cells and molecules is being forced upon the field. So really, that's all I can say. That I think we need more thought about narratives in general in science. But that's, as I said at the end of my talk, why it's great to come to a group like this, where what I also want to do is to think, well, what are the new, how do we tell this story in a way that gets away
Yes.
from those old paradigms?
Thank you. Thank you.
Totally. That was awesome. Kate?
Hi, Philip. It's great to see
Hi,
you.
Kate.
You're in good company, because there are several people in the audience here who are already offering a new ohm for you, but one that will really help move the narrative. And that is the concept of the genome, the collection of sensory capacities and how they interact through agency interacting with the environment, and how these top-down constraints interact with this bottom-up flexibility. I've been advocating a teleological narrative of, an overall narrative of self-regulation, because that's really what's going on at multiple levels. And it comes from the understanding that the emotional system is different and older and more informative and interactive with all of our regulatory processes, because it provides a very beautiful coarse graining of self-regulatory information through the simple experience and difference of pleasure and pain, what I call the fundamental semantic information bit. And there's ways that this fits into the computational story and all kinds of wonderful things. But it puts the organism and its agency, its decision-making capacities, and its intelligence center stage. It puts learning center stage, in terms of whether you want to think of it as model building that matches the environment, or if you want to think of it as the development of unique potentials, which are then variable parts in the social organism that we participate in. So what I think is really missing from the narrative and from our physics, the machine metaphor, is the concept of identity. What is it? How is it bounded? And yet the self, not self distinction, is writ large through our genetic and epigenetic and immune responses. But the self has gotten such a bad rap from selfish genes and biased this and biased that. I'm a huge advocate of not only restoring the organism, but the dignity and mystery and majesty of what the living organism does for us, that we don't have to think about at all. All of these beautiful processes that we're describing are what's in the unconscious realm of us. But all we need to bubble into our awareness is the simple distinction between something that's good for me and something that's bad for me, which is the very deep binary substrate of our emotion, the emotional, the information that's given to us by emotion that's been confounded within cognition. So there's lots to say. I have to bug out, I'm afraid. I've got something to present at 9:30, but Stu and I want to come and talk to you and hang out at your place in Wales.
Fantastic. Please do. It's wonderful at any time of year. Yes, you're always welcome. So, no, thank you so much, Kate. And that, I mean, what you say touches on so many things. I sort of hear resonances with them, that the idea of, of valency in responses to the environment, what is good and what is bad. It's this question of values. That I absolutely think is central, that we need to track back. I mean, again, people will become uncomfortable about, a the sense, or some people will become uncomfortable about the sense of a bacterium feeling good or bad about something. But I think, there's evolutionary continuity, right down to the level of single cells that are in some sense making these judgments of value. And they're making them not by looking up in some table, internally programmed table, is this a good thing or is this a bad thing. They're making those judgments with reference to their internal state at
And
that
their
particular
external
time.
state. And their
Absolutely,
external
yes.
state.
So
Yeah.
their relation to the environment, but also, but the relation to the environment, in a sense, is, something that, microbiologists are happy with. That you have these sort of interfaces, these receptor proteins at the membrane surface and so on. That's all fine. But the fact is that there is also the internal state, which, is not one might talk about it as an emotional state in a more or less metaphorical way, but it's certainly something that I think is evolutionary continuous, with an emotional state that we might experience, because it is a kind of state that is evaluating information within the context of the self. So I think that question of the self again is really important. I think that it's something that Kevin Mitchell and Henry Potter stress when they talk about the requirements of an agent. One of them is that it has to be bounded, that there has to be an inside and an outside. There has to be a place where the environment starts. There can be, there's obviously fuzziness there in terms of symbiotic systems and, nested symbionts and so forth. But nevertheless, I don't think that undermines the basic idea that there's some place where the agent starts and stops, and its environment sort of takes over. So I think all of those things are absolutely true. But I think also, Kate, you touched on an issue that I really think is important about this, which is there's an ethical dimension to it. There's an ethical dimension to recognizing agency not as an as if thing in biology that we talk about, but as a real thing that recognizes and respects the organism. And that's particularly important in these times when our own sense of agency is being undermined by ideas about genetic determinism and about determinism more generally and lack of free will. And I think a way of arguing from biology about the importance, the centrality of agency in our understanding of life absolutely does have an ethical dimension to it in restoring respect for the things we find around us.
Yeah, I just want to make one tiny follow-up. The feeling of pleasure and pain is connected to approach and avoid behavior. So you look at the behavior, which I tracked in a paper about how it works in the bacterium, the chemistry of it. But the logic of it is what's key. The evaluative piece, something that's good for me, bad for me, but it's also about identity, both parts and wholes. And it's also a reflection of the criteria for natural selection. You have negative emotions that are mediating self-preservation of form and memory and all that, but you have the positive emotions, which, by the way, we prefer. That's the true north of happiness and all of the complex positive emotions. That's about self-development, and that's the whole domain of phenotype, that we're building our own phenotypes constantly. So there's a direct connection between that sort of binary valence and the criteria for natural selection. So there's lots to talk about. And I can send you some of my stuff if you'd like.
Please do, Kate. I was going to say that. I'd love to see it. Yes, please do.
Well, thank you.
Thank you.
Stu?
Phil, that was an absolutely brilliant talk, and I hate you for it. It's just wonderful. It's just superb. So I want to bring up two topics for us to consider. One is the concept, I think you know it, Phil, but maybe all of us don't, that I've been calling for many years a Kantian whole. So in about 1790, in the Critique of Judgment, Kant said, thinking of organisms, uh, organized beings have the property that the parts exist in the universe for and by means of the whole. So we are all Kantian wholes. I exist because I have a liver and a kidney and a spleen and hearts and so on, but they exist because they're part of me. All living organisms are Kantian wholes. Not every complex thing is a Kantian whole. A crystal is not a Kantian whole. The atoms of the crystal exist whether or not the crystal exists. And a heap of gravel exists whether or not, I mean, the pebbles in a heap of gravel exist whether or not it's part of the heap. So Kantian whole is a very central concept. Now let's pause over that. It gives us a non-circular definition of the function of a part. So what's the function of your heart? It's to pump blood. But your heart makes heart sounds and it jiggles water in the pericardial sac. Those aren't the function of your heart. The reason that this holds is that the function of a part of a Kantian whole is that specific subset of its causal properties that sustains the whole. This is really important, you guys, because we've struggled for years about whether or not the notion of function is just in our heads. It's not. And once we've said that, we have a non-circular definition about the function of a part. Now, once you've said that, then this notion of a Kantian whole, um, let's talk about it. So uh, there can be nested Kantian holes. So, for example, let's take a prokaryote as a first order Kantian hole, but a eukaryote has
Oh,
chloroplasts and mitochondria.
I don't know if you can hear me.
Yeah.
My screen, sorry, my screen froze, but maybe it was just me. Yeah.
I think,
Oh, well,
I think it's okay.
Phil, I'm introducing the idea of a Kantian hole,
Mhm. Yeah.
where the parts exist for the whole.
Yeah.
Then there are nested Kantian holes. A prokaryote is, say, a first order Kantian hole. A eukaryote, second order. It has in it what were bacteria, mitochondria or archaea, and chloroplasts. We are third order Kantian holes. We're multicelled organisms. We have bacteria in our guts. So that's a fourth order Kantian hole. I suspect the entire biosphere is nested Kantian holes. Hold on to that. Dawkins does not have the idea of a Kantian hole. Once you have the idea of a Kantian hole and that the function of a part is that subset of its causal properties that sustains the whole, genes are just other parts, and they're carried along in evolution like any other parts. And now, with that in mind, let's come back to something that Charlie, you were talking about when you were talking about Darwin's finches and how everything changes when the beak changes. If you have a Kantian whole, selection acts at the highest level Kantian whole. For example, selection acts on us. If I happen to have a fitter heart, my kids inherit it. Selection does not act directly on my heart, right? But my heart requires that my liver works, and my liver existence requires that the lungs work. And this means something fundamental, Charlie. It means that selection at the level of the highest order Kantian whole demands that a coevolution of the interacting parts so they function together. That I think is the fundamental answer to the coevolution of all of the parts of the beaks and in us. I mean, why is it that our multiplicity of organs function together? Well, they all had to for us to continue to exist. So that is the emergence of this kind of coordinated functionality. And I think it's worth thinking that the entire biosphere is nested Kantian holes. Probably that's true. Next thing I want to bring up rapidly, I know you know this, Phil, but let me just say it. There's been work now for many, many years, including by me, on genetic regulatory networks. And it has turned out in the Boolean idealization that you rightly criticize, Phil, um, that these are dynamical systems and they can be ordered, chaotic, or critical. Critical is the edge of chaos. And we now actually know, well, it all started with Jacob and Monod saying genes turn one another on and off. So there's bistable circuits like A and B repress one another. So you can have A on, B off, B on. That says that different cell types should be different patterns of expression of these networks. And it works out that that's true. So we now know it's really true that different cell types really are high dimensional attractors. That's that's established. You can see it by looking at the patterns of expression of all 23,000 genes, and they're in different patterns on different cell types. That's now routine. There's now very, very good data that genetic regulatory networks actually are critical. Most systems aren't. Selection presumably got us to being critical. And part of the answer is, you guys, critical networks evolve gracefully. They have many of the properties that you speak about, Phil. That's not yet morphology, it's the dynamical behavior of it. And that it may be part of the answer about how evolution occurs. So I wanted to put those two things together. And then I want to make one more point and then ask you something. These well, intrinsically disordered proteins are not druggable by small molecules. And the reason is small molecules need a good binding site. And so part of a whole group of us is trying to think about how to make medicines for that by making peptides and screening them to find useful peptides to interact by binding to the surfaces of intrinsically disordered proteins. And I think you're absolutely right that the binary idealization is a good starting spot, but it's wrong. There's something about the fuzziness that you're alluding to and these condensates that are just fascinating, Phil. With that, I want to come to a last thing that you said somewhere, and it's the notion of potential function or affordances. So affordance comes from Gibson. A horizontal surface affords you a place to sit. So affordances are enablements. Okay, they're not causes. So the modem did not cause the internet. The modem introduced a way to share files, and that enabled the coming into existence of the internet. There's something fundamental going on with affordances, which are all kinds of mutual enablements. And Phil, you pointed to this notion about molecules with all of their fuzzy, floppy, what you were pointing at, as somehow kinds of affordances. It feels like there's something just fascinating in there. It somehow is related to the emergence of novelty in evolution. And I'd just like to ask you to be brilliant for a moment. What does all that mean?
That's thank you, Stu. That's precisely the question I want to try to grapple with. But one thing that occurs to me in that, and when you were talking about Kantian holes, um, the temptation might be then to think about a system, an incredibly complex system of cogs, where everything fits together and is working together for the whole, and each cog has a particular function within that system. But I think I feel clearer now, and I think probably what you're saying at the end there is similar to that. That actually the kind of Kantian whole that living things are is not like that. And I'm starting to think it's probably necessary for this sort of Kantian whole to involve the production of this, what I think of as adventitious matter, of these molecules and molecular aggregates that might do something, that have potential functionality. That seems to be.
I love it. Please build on it because this is where notions of attractors and dynamic so far we don't get. It's about
Yeah.
so please a fundamental thing is where does novelty come from in evolution, right?
Mm.
Please
Yeah.
just continue. I'm just getting excited. I'm sorry.
Well, yeah. Well, I mean, I think in a way, those are two probably related but different questions. And I think when you have a system of that sort that has components that don't have a particular function but that could find potential functions, that is a way where of getting novelty in evolution. They can find a new function even if they have one already. These moonlighting protein,
Yeah.
they can find another function that will create entirely new evolutionary possibilities. And in fact, perhaps that is the only way in a sufficiently complex system where you can get that kind of evolutionary novelty. But I also think that in any case, it's, in a sense, you could see that almost as a kind of an epiphenomenon of the fact that if the entity is to work at all as a Kantian whole of that complexity, then it will need to have this kind of looseness within it. It will need to have this pool of stuff that might do something in particular circumstances. It might, find a function, or it might find a new function with another aspect of that. The fact that we have so many of these small molecules, these low information molecules, not just non-coding RNAs, but microproteins as well, that, of which there are more and more. And I'm kind of kicking myself in that given what I've sort of said in the book, I should have seen it coming. That of course we would have proteins like this, or if you can call them proteins, we would have peptides like this, just as we have non-coding RNAs like this. That again provide this stuff that in combination can do something, even though individually their information content is very low. So I guess that's what I'd sort of, suggest adding to your picture, that that a Kantian whole like these ones that we are is not going to be a system of precisely intermeshing parts. It's going to have some of those, but it's also going to have a reservoir of stuff with affordances. And that's the only way it's going to have the versatility, the ability to innovate in behavioral terms, that it will need in order to exercise its agency.
Charlie, can I add one more thing briefly, please?
Oh, absolutely.
Well, once you have the notion of a Kantian whole and the idea that the function of a part is that subset of its causal properties that sustains the whole, the same part has lots of other causal properties, right? Your heart also makes heart sounds and jiggles water in the pericardial sac. Therefore, and this is a Darwinian pre-adaptation, or Gould and Verba's exaptation, the same part might find, and you already said this, might find some circumstance in which some other causal property is useful, and that then gets selected and a new function comes to exist in the biosphere. My beloved example is the swim bladder. Charlie, just forgive me for a moment. So the swim bladder is a sac partially filled with air and water. The ratio to its neutral buoyancy in the water column. Well, that's great, if you're a fish and you want to know if you're at the neutral buoyancy. But the way it evolved is there's lungfish, and paleontologists think water got into the lungs of lungfish, and the lung evolved into a swim bladder. Well, that's exactly the same structure here. A lung comes to have a novel function, neutral buoyancy in the water column. And I am absolutely convinced that open-ended evolution requires precisely that. And furthermore, you can't deduce from the fact that the lung functions as a lung, you can't deduce that it can be a swim bladder. So open-ended evolution requires steps that cannot be deduced, that therefore are not entailed. So a fundamental thing is what yields open-ended evolution. Biology does it. So far, artificial life has never done it. There's something fundamental going on.
Mm-hmm. Thank you. Thanks, Steve.
Fantastic. Thanks. Bill?
Um, I thought you did a particularly good job, Phil, and I enjoyed your presentation very much. I'd like to offer this comment. I particularly liked your slide that modern biology shows, and then you enumerated, I think, 6 or more different facets. And it struck me that these are exactly the components of cognition-based evolution. We had traded emails a couple of years ago, and I had introduced you to it. And I'm hopeful that you will consider a re-exploration, because every single thing that you write about is codified within that and enumerated in books and academic articles that are peer-reviewed. Importantly, put agency at the center, and the reception, internal interrogation, and deployment of information as the centrality of what cells do. It's always a cell-centered narrative, and cells solve problems. In such a circumstance, genes are tools. They're retrievable and deployable memory. They are solutions to previous cellular problems. So you have a single consistent narrative, and I think you might find it worthwhile to re-explore cognition-based evolution and find that everything that you've been writing about and those things that you're not quite sure how to put it into an entire narrative form is comprehensively presented and detailed there.
Mm-hmm. Thank you, Bill. Yeah, well, I'm absolutely happy to revisit that idea. I mean, what I would say, and I think maybe I did say, is that the cognitive narrative is one that I think is a much better narrative for thinking about these, but these biological systems from the molecules up. I think it's a much better narrative or metaphor for using than a computational one, for example. So I'm very happy with that. And Mike Levin and Dan Dennett's Cognition All the Way Down makes a lot of sense to me. I suppose where I'm unsure, and really it is unsure, is to what extent when it becomes more than a metaphor. And I'm not even sure in myself whether I mind too much, whether we actually call it cell-based cognition or whether we say we'll use a cognitive metaphor to think about what's going on. I'm not sure that those two are, in the end, are very distinct. Perhaps it depends what we mean by cognition. But I am absolutely, open to the idea. I'm absolutely enthusiastic about the idea that thinking about these systems as if they are cognitive, and probably systems that have this evolutionary continuity with systems that everyone will happily accept as cognitive, I think those are absolutely useful ways in fact, those are the best ways I see of thinking about them. And one of the things that I like about that, and one thing I say in the book, is that whatever these new narratives are, I think they are going to need to be narratives that come from within biology itself, rather than importing, technological metaphors to try to describe what is going on. So from that perspective, I think thinking about these systems in cognitive terms, I'm entirely happy with that.
I would just finish by saying František Beluška is also on this on this Zoom with us, and Catherine had mentioned the Seno. And František began that concept, and I've helped him with it, and we've partnered together to develop it. This, I think, offers an extremely robust way of looking at how cells approach their interface with the external environment and how they act as agents in a continuous internal endogenization of environmental cues. And we feel that that's a deep enough process to constitute cognition-based biology. The reception, analysis, and deployment of information, I think, is where the action in biology is going forward. I love your approaches as diminishing the prior gene-centric focus of the 20th century. I think we're in a new 21st century And I think biology is changing very rapidly. And I'm delighted with your presentation to see that you're right at the forefront. Thanks very much.
Thank you so much, Bill. I mean, I should just add, I know that Nick Lane is also interested in these ideas, and I speak to Nick a lot, and we'll be doing so again soon. So I'm really interested to discuss, those ideas with him too.
That's awesome. Thanks. As you've been talking, Phil, I've been taking notes of all of your friends and collaborators that you've mentioned, and I'm just so curious to chase down some of those relationships because it seems so um yeah, you're just like I love how you're at you're a sort of nexus of interesting questions.
Well, I guess I feel maybe if I have a role in all of this, it's to kind of put ideas out there and try to see, spot connections between ideas in different areas. I mean, originally I was a condensed matter physicist, and I feel like if I were still doing that today, I would absolutely be studying the condensed matter of the cell, which is, one of the most exciting things in cell biology. So yeah, I guess that's what I figure is the role I can try to usefully, pursue is making those connections.
Yeah. Yeah, but it's also like, I mean, that's a philosophical project, the idea of explaining and seeing the connections between things. You really have a great way of um an accessible way of like, exploring the philosophy of the facts that you're remembering and introducing. It's really inspiring and super useful. Yeah, so we're getting close to the end of our time, and anyway, I had another subject I wanted to bring up, but it's going to take too long, and so I think I might have to just
Uh,
leave it where it is. But yeah, just do you have any thoughts like as we close up and say our goodbyes? Do you have any closing thoughts or any ideas that have come to you as we've been kind of going along?
I guess I'd only say, and I anticipated this would be the case with, knowing who was, or some of the people who were involved in this group. Um, it reflects my growing perception, as Joanna, well, Joanna was saying, why hasn't the narrative? Why have we still got the same narrative? I think that is changing. I don't think it's. I mean, of course, everyone here will say that or will want that to happen, but I see that happening more broadly. Uh, more and more often as I talk to the guys, the molecular biologists who are doing stuff at the benchtop, making cell cultures and trying to figure out what the hell this protein is doing in this process, in this developmental process. The more and more I feel that I'm pushing at an open door when I talk about these sorts of issues and or ask the broader questions. Well, why are things this way? What does that mean? How does that relate to this? I don't get people hanging on to saying, well, ultimately it's all the genes or ultimately it's all mechanistic. There seems to me to be a real sense, a real sort of hunger for finding new ways of talking about these issues at the really at the benchtop level. So I am really optimistic actually that there is going to be some sea change in the way we think about these problems.
Totally. I totally feel that. I think a lot of us are just kind of waiting and not even waiting, but actively trying to make this happen. Yeah, and I just wanted to, welcome you to continue being a part of these conversations with us. And we so far I've just met about once a month, but we may do a little bit more in the future. And we also there's some talk of us having a conference and that sort of thing. So yeah, we're definitely all trying to make sense of the same very deep question all together. So
Fantastic. Well, thank you so much, Charlie, for asking me. It's great to be involved.
wonderful. Well, thanks so much. And thanks to all you guys. And I'll send the transcript and the recording soon. Thanks so much.
Thank you. Okay.
Okay. Bye bye. All right.
Hi, Charlie.
Hey, thanks for joining.
That was awesome. I mean, I listened to the whole
Yeah,
thing. It was amazing.
I'm gonna pause the recording now. Yep. Stop.